Извиняюсь, давно не пользовался. Так, теперь должна пойти трансляция уже.
Окей.
О, трансляция началась. Сейчас я ее замучу у себя. Так, hello everyone. Today I'm presenting you a workshop by Quantori team. My name is Maxim. I'm lead of Google Developer Student Chapter at Nazarbayev University. And today we're joined by Anna Koltakova. She's a PR manager of Quantori team, and Artem Kasianov. He's a PhD at bioinformatics and a senior data scientist at Quantori. Anna, please present yourself.
Okay, hi everyone. Thank you for attending us today. We are excited to meet you, and we are glad to see you as a participants of our ML hackathon. This is our first hackathon in collaboration with Nazarbayev University. And before we explaining the rules and holding our tech workshop, let me tell you a little bit more about the company. And please put plus in the chat if you have ever heard about Quantori. Maybe you joined us during Quantori week or attended job fairs in different universities. Please let us know if you have heard about us. And as plus, thank you. Who else? Okay, some of you already know about us, but for the rest, I'll give you an overview of Quantori. My name is Anna. I'm a global public relation manager. I have a huge experience in public relation, marketing, and communications, and I have been working in IT more than 7 years. And I want to ask you, tell about yourself, please, in the chat. Write a few words, for example, from which university you are or which faculty you represent today. While I'm speaking, I know that many of you from Nazarbayev University. Am I correct, Maxim?
More over the half of participants of our hackathon are from Nazarbayev University, and they represent different faculties like mathematics department, biology department, and mostly computer science departments. But some of our students are also from School of Medicine, so they represent molecular medicine department. Some of the students are even from KAIST University, some are from Inha University, so we have a very diverse list of participants on this hackathon.
Nazarbayev University School of Medicine. Great. This is interesting. Computer science, Nazarbayev University. Hi, Aldamjar from Nazarbayev University. Good. Nice to see that we can join many students from different universities. I think this is a really great opportunity for you to participate in such events, especially because Quantori work with life science and healthcare industries, and this is an extra opportunity for you to join IT company being not a representative of IT faculty, but having scientific backgrounds. So Quantori is end-to-end digital IT service provider for the life science and healthcare industries. And our aim is to help biotech and pharma clients to unlock the power of digital IT and transform life science industry and increase productivity of different possibilities. And what do we do? We help our customers to analyze data more effectively, to develop new medicine, and improve, hence, the quality of patient care. And Next slide. Okay. We work with different companies and from biotech startups to pharmaceutical companies, and we create our solution based on advanced scientific achievements. We develop algorithms and we process data from the management system for laboratories to genomic browsers. And we will share with you some examples later. Artem will tell you more specific information about it. Our offices is located in 5 countries. However, our employees work around the globe because we have remotely mode. We can combine to go to the office and work remotely, as it is up to you. And we have about 700 employees in our team. That includes software engineers, QA engineers, business analysts, data scientists, data engineers, and individuals with scientific background, as I said before. Regarding to our project, I can mention, for example, a system based on deep learning that detects COVID-19 and distinguished it from pneumonia using patient chest X-ray images. And also another example, Quantori created a model that analyzes colonoscopy images and predicts the presence of cancer, tumors, or other diseases based on just these images. The headquarters of the company is located in Boston, and we have another offices in Tbilisi, Yerevan, and Lisbon in Portugal. And we opened our branch in Astana last year. So we're going to celebrate our first anniversary in Kazakhstan this summer. You can see this is a short list of our clients, and mostly we work with clients from USA and Europe, and you can see that reputable famous companies trust us and work with us, such as Pfizer. I'm sure you know this company, or another companies, for example, life science providers such as Perkin Elmer or LGC project. This is international life science company. Also, we work with venture funds as flagship Pioneering. This company invests in biotechnology, life science, and health. And also, as an example, Jackson Laboratory, JAX. This is research institution, this independent research institution. You can see our key expertise area. We have been working with pharmaceutical and life science industries for over 2 decades, and we provide our customer with excellent service and modern solution. And for this, we use our huge expertise and knowledge in these areas. For example, we designed and deliver ML solution to create deep learning models that recognize and predict data patterns with more accuracy. And Artem will tell you more about this. Or we develop data science, digital engineering, technology platform. Also, we work with cloud transformation projects, DevOps, and other solutions connected with cloud services. On this slide, you can see our tech stack. And I can tell you a little bit more about benefits for employees. As I mentioned before, we can work at the offices or remotely in any location. So also we have flexible working hours. This means that you set your work schedule as you want, of course, according to your customer schedule and by approval of your manager. Also, you have an opportunity to have one-on-one English lesson to improve your speaking and writing communication. Quantori provides employees with healthcare benefits such as medical insurance, day offs, and paid sick leaves. And you can get certification paid by the company, and you can opportunity to educate, to improve your soft skill and tech skills, because we have a lot of professional educational program for our employees, for current employees, and for external audience, as we have our own corporate university, Quantori Academy. It was founded in 2020, and 2021 we launched external IT courses for people who want to join IT or change their career path. And since that, we have received more 3,000 applications, and I believe more than 300 students graduated from Quantori Academy. So the main benefit is practical knowledge, because teachers are experienced specialists from our company. They are current developers, so they share not just theoretical materials, but also their real cases from real projects. So this is a really good opportunity. I'm sure you need to start career in IT, because you agree that very often when you search vacancies, you see job position with requirement for experience, senior or middle positions, but you're a student and we understand this, so we are ready to teach you. There are some external schools, JavaScript, Python, QA, but JavaScript, Python, and QA are already going. And perhaps we will start DevOps and data engineering school maybe next quarter. So for this is Quantori benefits. Yeah, this is absolutely free of charge, and this is absolutely remotely. You need just maybe from 8 to 9, 10 hours in a week to spend to do home tasks and to attend classes, online classes. And please follow us on social media to be tuned, because all courses are announced in our social media accounts, not to miss this opportunity. Instagram, Telegram, and LinkedIn, what you want. And if you have any question, you can contact me anytime by email or just message me on Telegram. Thank you.
Thanks, Anna. Now let's do a little interactive. So I want to ask our chat, what is your favorite tech stack? What do you use for your machine learning tasks? Is it sklearn or PyTorch or TensorFlow? You can just type in the chat. We'll see how you vote, what's better for you. And now we can go to the presentation by Artem Kasianov. He'll tell us about his expertise in disease detection.
Yeah, thank you, Maxim.
We'll start a little bit with your introduction. You can
okay. I need to say about me. My name is Artem Kasianov. I am a senior data scientist and informatician in Quantori. I have a PhD in bioinformatician. I got it in I think 2 years ago. And before Quantori, I worked a long time in the academical institutes and do some science. Yeah, now I'm working in IT, and Okay. What I want to tell you in my lecture, in my presentation, I want to convince you that we can use DNA to diagnose diseases. Yeah, because it's not obvious fact. Yeah, and I want to show you how we can use it, why it's useful for disease diagnostic. And my own interest, I provide for this hackathon data, and I'm very interested in the results with this data, how it works for diagnostics. Okay, let's begin. Yeah, it's some fun picture about what is diagnostic of disease by DNA. I think some of you, maybe most of you, heard about the genetic tests. Yeah, and about the genetic test industry. And it's not my own idea about this industry that, okay, genetic test is very good for detecting, for diagnostic rare diseases. Yeah, but for complex diseases, it may be not so good because there are a lot of factors. And the companies who try to do genetic diagnosis in most cases try to convince us that we can use genetics to predict not only diseases, we can predict, for example, I heard about the test that we can predict for children what sports they need to prefer. Maybe they will become a better footballist, or they can better play in the hockey, and it looks like it's fortune telling. And I have some concerns about working of this test, but may happen because we don't have enough data, and I will tell you why I'm about my concerns and why people think they can use genetics for this prediction. Okay, and this slide you can see the name of the companies. There are a lot of companies which try to do the genetic tests. There are a lot of companies in the world who try to do it, and I think the most famous company is 23andMe, and I can say that most of companies it's not idea that companies for genetic tests begin from the disease detection. Yeah, it's the most useful thing for genetics tests now is try to detect some genetic disease. But most popular product from this company isn't the disease detection. It is people want to know the ancestry, want to know the nationality, ethnic, each own ethnicity. That's why people try to pay the money for these companies and try to do the test about the ancestry. Okay. About their ancestry, yeah, and I think this is the most popular product for these companies. Okay, next, let's begin to speak about the biology. And first idea, yeah, all of these companies try to do genetic tests by using DNA, yeah, but we can try to think what's the most what's the part of our bodies is the most important, what's the most important molecule in our bodies. Yeah, not the most important molecule, big molecules that do every work in our body. And this molecule isn't a DNA, it is a protein. Proteins in our bodies do the most of work. There are a lot of functional proteins, and one of them, for example, protein is the components of the factory to create a new protein. So that it's a part of ribosomes. That's why protein, it's very important. That's why the people who humankind, before when they try to study live organisms, they begin from studying proteins. Yeah, and I think it's a good idea, because if we have a disease, yeah, we need to understand what's the cause of the disease. Yeah, it's some malfunction in our body. That's why we have some problem with some protein in our body, and we need to find the incorrect protein and to repair it, for example. That's why we need to understand what the protein isn't. What protein is bad, yeah? Okay, that's why it's very important to understand, to know what is the protein. Yeah, on the slide, you can see some ideas, what is the protein. On the right side, you can see on the right side, on upper part of the side. Okay, I need to say, what is the protein? Protein is polymer molecule. It's a big polymer molecule, and each polymer molecule consists of monomers. Yeah, and in the upper right side, you can see the structure of monomer. It's amino acid. And there are 20 different amino acids in the live organism proteins. And all of these amino acids differs from each other by the side chains. Yeah, it has the same amino group and the same carboxyl group, and has different side chains. And in the protein, this amino acid connected by using peptide bonds. Yeah, it's the main idea about the peptides and how people try to study proteins. They study the different types of structure. First, the most simple structure of protein is the primary structure. It's a simple sequence of amino acids. Looks like some board, something like that, and it's only a sequence in the 20 letters, not 20 letters alphabet. Yeah, it's very simple structure. But in the live organism, protein folded in the some 3D structure. And to understand the function of the protein, we need to understand the 3D structure. Yeah, and this is the last quaternary structure. And scientists first try to study the 3D structure of proteins. It's okay, how we can do it? What is the method we have to study quaternary structure of protein? Okay, the most oldest method is X-ray crystallography. It was invented in 1914. It's a very old method, and today it used tool. Yeah, and how it works? First, we need to extract protein from the cell, from the live organism, and create the crystal from these proteins, and do the X-ray, measure electron density map, construct based on it atomic model, and in result we have this beautiful picture, yeah, of the protein. But I need to warn you about this method. First, it is structure of the crystal. Yeah, to create the structure, we need to extract protein from cell. During this process, we lost the in vivo structure of protein. What it means? We measure the structure of protein, isn't it in the live cell? We change it. Yeah, that's why it's very maybe it's not correct to say the structure is incorrect, fully incorrect. Yeah, but in some cases it's a change structure, and in a live cell you can see another picture. And another problem with this method, it's not a throughput. Yeah, it's not, you need a more, it's very labor-intensive method. Yeah, and to study only one protein, you need to many time. We need a lot of time to study only one protein. That's why it's very difficult to study all of proteins. Why? I can say in the human genome, for example, we have near the 30,000 of genes. Each of these genes has its own protein, and we need to study all of human proteins. We need to study 30,000 of crystals. And we need to do the 30,000 of X-rays, and it's very difficult. It's labor intensive, and it's not cheap. Yeah, that's why this methods can give us good result, good fast result. Yeah, it can give us a result, but it's not fast. Yeah, and this is the example why it's not effective. As you know, the X-ray, we know X-ray from the beginning of 20th century, and in PDB, it's a big database of the protein structure. We have only near the 200,000 of protein structures, and we created it in 100 years. Yeah, it's very small amount of data, because we have, for example, example about the, not for human, not for only for human, for example, for insects. We have thousands of species of insects with different proteins, and each of these species has thousands of proteins. And we have I think in the nature we have millions, maybe billions proteins. Yeah, and if we need 100 years to study only 200,000 proteins, I think we can't do it. We can study all of proteins. That's why we need another method. Okay, every lecture about using bioinformatician, using machine learning and bioinformatician has this slide. Yeah, the slide about the AlphaFold tool. It's a very effective example of using machine learning in bioinformatician. What is this tool? Its tool predicts the 3D structure of protein. It predicts quaternary structure by primary structure. And it's a very big achievement, because you can see on this slide, you can see this graph, this plot. On the horizontal axis is different methods. Yeah, and the smallest bar for AlphaFold. Why? Because on vertical axis we have information about the error. Is mean error of reconstruction for some test set. Yeah, and we can see that the AlphaFold reconstructs the 3D structure with very high accuracy. Okay, we have a method that takes the sequence, primary sequence, some words in amino acid alphabet, and we can reconstruct the 3D structure. Okay. And we speak that it's very important thing. That's why we somehow generate a huge amount of primary structure. Yeah, that means we can't study 3D structure quickly, effective, with high throughput, but we can somehow find the primary structure of proteins. How we can study primary structure of proteins? How we can find the sequence of proteins? Okay. First and oldest method for this task is to do the mass spectrometry. as protein mass spectrometry. Is this method used for study protein sequences? Yeah, the main idea of method, you have some sample from live organism, you extract proteins from here, then put it into the mass spectrometry. In result, you have the masses of the proteins, and you can identify proteins in the sample. Yeah, how it works? First, you have cells with protein mixture, then you extract protein, then you need to digest proteins into peptides. What is peptide? It's a small sub-sequence of protein, so small sub-proteins, yeah? And after that, in result of mass spectrometry, you have masses of the peptides. And by analyzing those data, you can understand what peptides you have and what amino acids you have, and you can somehow reconstruct the source proteins. But what's the problem with the mass spectrometry? Why it's not main way to study primary sequence of proteins? Because peptides and some amino acids may have this very similar masses. That's why you during reconstruction, you may have ambiguous peptides. You can't reconstruct protein uniquely from the mass spectrometer data. That's why to effectively use mass spectrometry, we need a database of known proteins. Yeah, that's why we need another method to study proteins, because we can do it by using protein mass spectrometry. Okay, and on the slide you can see the contents, the number of proteins in the one of the biggest databases or protein databases, UniProt. Yeah, and you can see that the humankind know more than 200 million proteins. Yeah, and as you understand, we can't use to solve this task. We can't use X-ray and we can't use mass spectrometry. How we find this big amount of proteins? Okay, and I need to show you the main slide. When you try to tell something about the molecular biology for non-specialists, you need to start from this slide. Yeah, but I want to I try to show you why it's important. Yeah, before we can discuss it. What happens here? Yeah, another main component of life organism is nucleic acids. Yeah, DNA and RNA. And main function of this molecule is to store genetic information. Yeah, and how works the genetics in the cells? Yeah, first we have genetic information stored in DNA, then we transcripted it into the RNA, and protein created by using RNA as matrix. Yeah, and all information about the proteins stored into the DNA. That's why we can study DNA to understand all of proteins in the live organism. We don't need to extract proteins to study structure or to do the mass specs for different organisms. We simply need to study DNA to find the primary structure of proteins. Okay, and next, I will tell why we think that the study DNA is better. Yeah, first I need to say a few words about the DNA. Why is DNA is useful for storing genetic information, because it's very stable structure. First, it has it's double helix structure. Yeah, that's why for each of DNA molecule we have 2 copies of information. And first idea why we need 2 copies. First is we need it for replication, for copying, because you know about the cell division. I think you know about the cell division. When cell divides, it creates 2 new cells. Yeah, and for each of cells we need DNA, the similar DNAs, and that's why we can split this double helix structure, and by using the special proteins, copy the second helix, copy the second strand by using the first strand as the matrix. Yeah, and another idea that the cell may number of errors may be okay. In the time we have a DNA, yeah, and there are a lot of environment factors that can affect the DNA, and it can add some errors to the one of the strand, and but we have another strand, and we can use this information to repair DNA. That's why it's very important to have the second copy. Yeah, and that's why DNA is very stable. And another idea, another example. Maybe you heard about that the scientists can work with the ancient DNA, can extract DNA, for example, from the dinosaur bones. Yeah, and they can do something with this DNA. It isn't as in the Jurassic Park film. We can't reconstruct dinosaur, but we can study it. We can study some mutation, some difference with modern birds, for example, because the most similar organism to dinosaur is birds. And what's interesting, the DNA in the bones of the dinosaur we can extract it and we can analyze it, and this DNA preserve information. Yeah, it's not lost. It may bones may have the 1,000 years and something like that, but DNA here partly intact. Yeah, you need to understand that it's not the same DNA when this dinosaur is running around, but we can extract useful information from it. It means DNA is very stable thing, and it's very good storage of information. Okay, and another main idea that in the DNA you can find the genes. Genes is the records of proteins. And what happens? How we study DNA? We can do some sequencing. I will show you a slide with the sequencer. Next slide, and you can find the text, the big text in 4. Oh, okay. I don't say that there are only 4 letters in the DNA alphabet. Yeah, and when you study DNA, you have a very big 4 alphabet text, and based in this text you can find the words. What is the genes by using some machine learning methods, and the genes you can translate to proteins. You can computationally translate by proteins. It looks like in the cell. Yeah, in the cell, what happens in the cell? The genetic system of the cell finds the gene in the DNA, transcript it into the RNA. It's a special molecule. Transfer the RNA to the protein factory, name of this protein factory is ribosome, and ribosome creates the protein. And here, two codes. First code is how to convert DNA string to RNA. It's very simple. 3 letters is the same, but T letter we need to replace by U letter. Okay, and then RNA alphabet we need to translate to protein alphabet. It's in the RNA alphabet you have 4 letters. In the protein alphabet you have 20 letters. How to translate it? It's translated by the it's a third 3-letter code. Yeah, each 3 letters in the RNA encodes 1 amino acid, and you can see here, for example, how to understand it. Okay, this is the first letter, second letter, and third letter, and you can see that GCA is alanine, GCG is again alanine. For example, GGA is glycine. Yeah, and how it works. You can, and computationally, you can take the genes from DNA and translate it by using this genetic code to protein. In result, we have, okay, how we can study DNA. The first device to study DNA was invented in 1977. It's 3 years before the protein mass spectrometry appears. Yeah, and again, it isn't a very high performance thing. It's very slow method. And after that, people try to study proteins. Yeah, because we have a very slow thing to read the DNA, and we can read a lot of DNA, and we have a very fast mass spectrometry, and scientists try to study proteins. But in the beginning of the 21st century, humankind develops high throughput sequencing. It's the second generation here. And now we have a very effective and very fast technology to read DNA. I have a third generation now to read DNA. It's very fast development field. And here's a very famous slide. It's about the cost per human genome. What it means? It's the cost you need to pay to sequence your genome. Yeah, and here, at the beginning of sequencing era, next generation sequencing era, to sequence a human genome, you need $100 million. Yeah, it's the cost of first human genome. It's near $100 million. It's a very big project. There are a lot of people who try to sequence, who did the sequencing. And this was a big achievement for humankind. But now we need only $1,000 for sequencing human genome. And as you can see here, the cost drops faster than Moore's law. Yeah, and it's a big achievement. But at present time it stops. Maybe it's a limit. Yeah, but I don't think so. I think this is a problem with the pandemic or something like that. And the third generation become a more powerful and more cheapest generation of sequencing technology. I can say that we have a sequencer that can be plugged into the PC by USB, and it's a very small thing. Yeah, it's looks like a flash card. Yeah, it's very small, and we can plug it on PC, and it can sequence a genome. but today it can sequence only a genome for bacteria or virus. But I think in the future, this device can sequence a human genome and do it not a week. And you don't need a week to do it. You can do it for 24 hours, for example. And it's very interesting device, and that's very perspective technology, Oxford Nanopore. Okay, and another story. Yeah, you can see here the decreasing of the cost of human sequencing, and on this slide you can see the increasing number of human genomes. On the horizontal axis you can see the years, on the vertical the number of known human genomes. Yeah, and here the dots here is different projects. For this project, it's a big project. It's a big project of several scientific institutes from different countries. What they did, they gathered together and take the blood from thousands of people and do some whole genome or whole exome sequencing and create a big database of the genomes. Okay, why it's important, I will tell you further. Okay, first I need to say you about the human genome, human karyotype. What is karyotype is a set of chromosomes. Chromosomes, okay, what is the chromosome? It is a part of human genome. Human genome is not a one text. It's 23 texts, and each part of the text is the chromosome. Yeah, and what's another interesting thing for this text? We have 2 copies of this text, but it's not the identical copies. It's not the double helix structure, but it's 2 chromosomes, 2 small different 2 chromosomes with differences. Why it's important? Why it happens? Because one chromosome we have from our mother and another chromosome we have from our father. Yeah, and in your genome you have a part of genome of your father and part of genome of your mother. Yeah, and it's very important for evolution. Yeah, it's some evolutionary mechanism. Okay, that's why. What is studied is genetics. Yeah, genetics try to find relationship with the genetic traits, with the different changes in the chromosomes, and try to correlate it with phenotypic traits, with the diseases, with the color of eyes, or something like that. And each genetic trait can be represented as 2 alleles, as 2, for example, letters or something like that, from father and from mother genome. That's why we can split it in 2 types. We can homozygous traits, genetic traits, and heterozygous. What means homozygous? At when on the one chromosome and another chromosome we have the same state of genetic trait. Yeah, and heterozygous when we have a different state of genetic trait on this pair of chromosomes. Why it's important? Yeah, when we have homozygous trait, yeah, you need to understand that here we have some protein. Yeah, and each chromosome can used to generate the proteins, and you have the same proteins generated from father chromosome and from mother chromosome. But in heterozygous state, you generate from father chromosome one type of protein, from mother chromosome another type of protein. and you have in your cells proteins from with small differences, and it may affect phenotypic traits. That's why when in the biology studied for each genetic trait, we studied 2 states from father and mother chromosomes. Okay, it's why it's important. I will tell you further when we will discuss the data, because here we have the states homozygous or heterozygous for genetic traits. Okay, and this is some milestone, and I think I can answer on the question. Oh no, I think let's discuss it after lecture. Yeah. Okay. Okay. What we need to know: we have each human has a DNA, and as we understand, each human is unique. That's why we want to expect that DNA of each human is unique, but we need to prove it. How we can prove it? Okay, we can prove it by application of genetics. Yeah, and the most oldest application of DNA is to using this DNA in criminalistics. Yeah, here there's some history about history of criminalistics, about using different science methods in criminalistics. And you can see that DNA fingerprinting was first used in the 1985 year, and we will discuss how it works. Okay, first we need to discuss how we can use DNA as fingerprint. Why it works? Because DNA is unique for each human. And I think I can say that difference is not so big. Only one mismatch on 1,000 nucleotides. It says here only 1/10 of 1% of DNA differs from person to person. Yeah, and for criminalistic we can use this information. And for interesting, as you understand, to use for criminalistics, we can't use the sequencing. Yeah, it's very expensive for criminalistic. We need to do a lot of analysis. Yeah, and if we need, you need to remember we have a 1990s, for example. Yeah, and the cost for human genome is near $200 million, and to do one analysis we need $200 million, and it's not a good idea. How we can study DNA? First, what is why we need DNA? What we want to understand from DNA? First, it's a criminalistics. We can identify a victim or link exclude suspect to crime scene, link multiple crime scenes, and the most famous thing is establish paternity. Yeah, okay, how we can do it? How we can do it cheap? First idea is to use the special proteins, restriction enzymes. How it works? We have a DNA, and we have special proteins that can cut DNA in the specific places. Yeah. And we can use this DNA to generate the fragments. And different people will have a different size of these fragments, because people have difference in the DNA. That's why they will have a difference in the restriction sites, because we have we have a special unique restriction sites, and each restriction enzyme can cut only unique restriction site. And if we have some mutation, some polymorphism in the restriction site, it will not work. That's why we will not have a special fragment. And each DNA sample from different people will have a difference in the sizes of restriction fragments. That's why we need only compare. We need only to okay, it's interesting question. Okay, what we have in result? We have in result different fragments, and to compare samples from different people, we need to somehow to sort these fragments by size, and we can do it simply by using the special porous gel and electrical field. How it works? We can put the DNA into the gel, then add the electrical field, and DNA try to move to move to anode, to positive charged end. The positive charged end. Okay. And what happens here? If we have a small DNA fragment, it can move faster. And if we have a big fragment, it will go slowly. And we have this picture. The biggest fragment. For biggest fragment, we have this light. For smallest fragment, we have light at the end of this field. And for different samples, we have this picture. Okay. Oh, and this is a more better scheme about it. We have DNA. We use the restriction enzymes, put it into the gel, add the electric field, and have the gel picture. Gel electrophoresis. The name of this method is gel electrophoresis. And let's try to use it for two cases. First, for identify a victim, and for paternity test. Here we have a result of DNA samples for crime scene and for 3 suspects. And we need to compare the crime scene, DNA from crime scene, with the suspects by comparing these pictures. And what we can here, I think the most similar is the suspect 2. And this is how it works. And for paternity test, we have data from mother, from the child, and for 2 male. And you can see the child has the similarity with the mother and similarity with one of the male. I think this child is the second male. Okay, and this is very simple. And this is very cheap. And we can use it for this task. That's why this paternity test isn't so expensive as sequencing DNA. Okay, what we have here, now we know that the humans are very different to each other by DNA. That's why maybe we can use this information to compare patients, compare people with the disease and without the disease, and maybe we can find the markers that can explain the disease, and try to, maybe we can use it to do diagnostics. Yeah, because it's very simple to extract a DNA. We can, for example, take the blood, or we can take the hair and extract DNA from there. It's not so difficult to do it. Okay, and here's a slide about the motivation. Why we need to study this difference between different people. It's name of this field is study human genetic variation. Human genetic variation studies. Yeah, people, scientists, try to find all possible human genetic variations. Okay, and what we know about the human genetic variations now, that the human genome has approximately 10 million polymorphisms, 10 million differences. And the most important type of polymorphism is a single nucleotide polymorphism. I think why it's important, because it's the biggest density, biggest resolution, biggest resolution of the genome. And we can, using by only one changes in the genome, we can detect, we can do some association with disease and something like that. And you need to know, if we have only one changes in the DNA, in the protein coding region, in the gene, we can change it. Maybe changed the protein, and changed protein may have maybe malfunctional. Yeah, protein may lose function, something like that. Okay. Okay, it's the same. I think it's the same. No, okay. There are a lot of different numbers. That we have 10 million SNPs with the frequency at least 5% in human population, and 5% is one of the important biology threshold, because the biologists like the 5% threshold. I don't know why the 5%, but it's some empirical threshold. And if you do some biological studies and you do some paper and you need to choose a threshold for some probabilities or something like that, you will choose 5%, and you publish paper with this threshold, and there are no answers. Not answer. No, there are no questions about it. Yeah. Why the first 5%? I don't know. Okay, and SNPs as changes in one nucleotide, only one nucleotide. There are a lot of different. There are a lot of other types of genetic variations. Yeah, for example, there are deletions, insertion, inversions, complete number of variations, and you can see it on this slide. But why single nucleotide polymorphism is the best genetic variation? Because it's very easily to study by using modern sequencing and other genetic technologies. There are a lot of such genetic variations, and they can easily associate it with the traits. Okay, and okay, about the SNPs. Another idea that the SNPs is correlated to each other by position. What it means? It means that the SNPs that near to each other are correlated. How it works? Okay, example. we have the father DNAs and mother DNAs. In father DNAs, we have as 2 chromosomes, and in mother DNAs, we have 2 chromosomes. And further, we have a child's DNA. Each of child's DNA is some combination of father's chromosomes. One father chromosome in the child DNA is the combination of 2 father's chromosomes. And in mother DNAs, it's a combination of mother chromosomes. How it works? It works by some special genetic mechanism. Name of this mechanism is recombination. Yeah, during the meiosis, during the cell deletion of human cells, we have this process when chromosomes shuffling, or the source chromosome shuffling in the new cells. But what's interesting here, you need to understand that nearest regions in the DNA, nearest in the child. That's why if we know one SNP in the middle of region, we can correlate it with the SNPs around of it. Okay, and it's very important because there is an important idea. Yeah, it's very difficult to study all of SNPs. Yeah, all possible SNPs in the human genome, because human genome size is near 4 billion nucleotides. It's very big. And we don't have enough people to study all of SNPs. Yeah, by direct way, you can see on the left side, on the left picture. But we can study, but if we have some correlation, if we find in our analysis another SNP, we know that we can correlate it with the SNPs near of it, and maybe by our analysis, why it's important? Because it's very difficult. Yeah, okay, next slide. Yeah, it's very expensive to generate thousands of human genomes. That's why for such studies, when scientists try to associate genetic variants with traits, with the diseases, they try to study not all of SNPs. They try to extract, for example, 5 million SNPs from human genome and try to find association between the 5 million SNPs and the trait. But it's not important to find this SNP that changes the protein. We can simply find the SNP that location is near the SNP, casual SNP. Yeah, the SNP that changes protein. Okay. And about the technology. For the studies used microarrays, microarrays. Yeah, it's some technology that we can study not on genome, the full genome. We can study only specific SNPs. How it works? We have some very big array with the DNAs, with the specific DNAs, and we can take the DNA from the sample, share it into the fragments, add to our chip, and if in our chip we have the region similar to the region in our DNA, it will create hybrids, double helix DNA structures. After that, we can use the special technology to find in which places of chip we have the hybridization. Okay. and we can understand what SNP do we have in the our sample. Okay, and there are a lot of works that try to, you need to know there was a lot of works. The main aim of this work is to study SNP, association between SNPs in human genome and some traits. For example, diseases or weight. Weight, it's a better idea. I think the height. Height is one of the most famous genome-wide association study is the height association study. Yeah, and I think we can predict the height by the DNA. It works. And here's some another problem with GWAS, with the genome-wide association studies. It is on this plot. This plot you can see the relationship without the number of cases, number of samples in the association studies, and number of genome-wide association hits. Here, and you can see that if we want to find new genes associated with the trait, we need to take more people. We need to spend more money. Yeah, and another problem that we can't do. We will discuss the statistic. Next, I will discuss the statistic, and I will tell why we can't use. It's very difficult to use full genome sequencing for this task. Why we need to use microarray? Why we need to use small subset of SNPs to detect something. Okay, and here's a slide about how grows the database with SNPs. There are a big database with SNP, and you can see in around 2012, there are a very fast increasing of number of studied SNPs, of known SNPs. This plot with the dots is submissions, and with the lines is ref clusters. Ref clusters means you have a submission of a new SNP, and after checking, submission uploaded to the BSNP database. And we can see that now we know more than, I think, 1 billion SNPs now, because it's old plot. I can't find it's new, because I think we have much more SNPs now. And what I can say about it, that why it happens, because there are a lot of GWAS studies, and most of GWAS studies happened around 2012 year. After that, it was stopped. Yeah, there are, at the present moment, it is not. There is not a big project about it for human genetics. There are a lot of projects for plant genetics, for agriculture, animals genetics, not for human genetics. Okay, about the statistics. Yeah, small story about the C-squared, how we can study association. It's very simple case. Yeah, we have the contingency table when we have two conditions. For example, we want to find association between the disease and the smoking non-smoking patient. We have this table, and we can use for this table p-squared test. And after that, we have p-value. Use the 5% threshold for p-value, and if the p-value is smaller than 5%, we have association. It's a normal situation. Okay, if we can change situation to genotype level, and we can encode cases, controls, and different alleles, combination of alleles by these definitions. And again, we have the same contingency table. I can use chi-squared. And after that, if you read something about the glasses, about the genome wide studies, you saw Manhattan plots. What it means? On the horizontal axis, you have a position in the chromosome, and chromosome, and on the vertical axis, you have a p-value in logarithmic scale, and if negative p-value, yeah, and we can see position of the SNPs with the smallest p-value, and we can choose this position as associated traits. Yeah, but the main problem of it that we have a huge amount of positions. Huge amount of positions, and that's why we need to add some multiple text correction. Yeah, that's why we need to choose some threshold here by using multiple text correction. And if we have all position in the genome, this correction can kill all of our results. It happens because for full genome, we have a very small amount of participants in our tests, because we need more people to have more hits in the more dense tests. And for now, why it stops? Because we don't have enough participants. Yeah, that's why it stops. Okay, about the results. During this glass, we have a huge amount of SNPs associated with the diseases. But why it's not enough? Because we don't interested in the rare diseases. It is not because it's not interesting. It's why because we know what happens in the rare diseases. Yeah, if disease, the cause of disease is changed in one protein, it's everything fine. We know that if we have the SNP in this protein, human will have a disease. Yeah, it's normal. But for multifactorial disorders, it's for some autoimmune diseases. We will discuss now inflammatory bowel diseases. We have another situation. Why it happens? For example, here's some example. It's some pathway in the immune system. Each of these, you can see a lot of proteins, protein complexes here, and what happens is some receptor, is some protein that can receive some signal. Yeah, and if we have some changes in this signal, in this protein, we can there are two situations. If we have changes in the genome change one of the protein in this complex, and this complex isn't works, this cell doesn't have a signal and doesn't work. If we have another changes in this protein, it may have more signals. It may react more intensively on the signals, and it may cause another problem. For example, in autoimmune disease, what happens in autoimmune disease? The T cell can receive more signals than it needs, and in result, T cells reacts on the health tissues in the human. For example, in inflammatory bowel disease, in result of this disease, we have that our immune system attacks the colon, and in the psoriasis, it attacks the skin, and it's a problem. And as you can see here, there are different arrows. Arrows to the different proteins, and in the genome-wide association studies, scientists found the changes in these proteins associated with these diseases. And you can see that there are a lot of small changes in different proteins may happen, and it affects this pathway. And we don't know a single way how these changes can happen. These changes can happen. That's why it may happen in many different ways. Changes in different proteins can cause the disease, and it's a problem. That's why I think we don't have enough information to predict diseases with big reliability. But for inflammatory bowel diseases, I can talk about it now. We have a huge amount of data now, and I hope we have possibility to predict it somehow. But in our task, we don't need to predict to classify people with disease and people without disease. We will. Our task is to differentiate between two IBDs, between two inflammatory bowel diseases. One is the Crohn's disease, another is ulcerative colitis. Why it's important, I can tell you further. First, I need to say the difference. Difference is very big difference between these diseases, because ulcerative colitis happened only in the colon. And that's all. It affects only colon. Immune system attacks the colon, and you can remove the colon, and disease will disappear. Crohn's disease is another thing. You can't remove the affected region because why? Because it affects all digestive tract. You need to remove all digestive tract. It's impossible. People can't live without digestive tract, without full colon, without another organs, and it's a problem. That's why it's very different diseases. But if you check the symptoms are the same. You can't differ it by the symptoms. And how works the differential diagnosis now? First, it's blood tests, different imaging studies, and the main stage is endoscopy, is colonoscopy. And by colonoscopy, you can find the difference because it's very different inside the colon. But it's very, it's not good procedure because you need to take a tissue from affected region. It's not good for patient. That's why we need a more gentle method to do differential diagnosis. Maybe colonoscopy, we need colonoscopy. I don't say that we don't need it, but maybe in some, when we have a big inflammation, we don't need to take tissue, and to increase the symptoms, and it's not good for patient. Okay, here I can say that we know much about the inflammatory bowel disease. There are a lot of glasses about it. That's why people, by using these glasses, find a 300 very good SNPs that can differ health people from people with IBD, and create a very small microarray chip for 300 SNPs. It's very cheap thing, and it's very interesting. Can we use this chip to do differential diagnosis? Because it's very cheap diagnostic technology. That's why we have data sets with the data from these chips. It's European population. There are 1,000 and near the 200 Crohn's disease patients, 800 ulcerative colitis patients. It's in the trained data set, and result, after you have the model, you need to predict the state for 20 Crohn's disease patients and 20 ulcerative colitis patients. Okay, and what is the data? First is the clinical data. It's some information about the patients. We have here the gender, height, weight, diagnosis, age when diagnosis was obtained, smoke status, and laboratory values for leukocytes, hemoglobin, C-reactive protein, and something like that. It's the set used for detecting inflammatory bowel diseases and inflammation. It's very simple values, and genotype data. It's for near the 400 SNPs. We have genotypes for each of the patient, and genotypes coded by 3-letter code. I can show you this code. First, we have 2 types of homozygous. It's and it's coded by -1 and 1, and one type of heterozygous. It's coded by 0. And you need to use this data to differentiate Crohn's disease and ulcerative colitis. Yeah, that's the task, and to create a diagnostic system for it. Okay, that's all, and I can answer only questions, and there are some questions here. I start from the last about my participation in Kaggle competition. I'm not participating in Kaggle competition on prostate cancer, but I heard about it. Okay, how do you think will it possible to crossbreed dog and cat in future? It's represented in cat dog cartoon. It's not in classical way. It's impossible. Yeah, because it can be crossed. Because there are a problem with the animals. Yeah, with animals we have a problem. We can cross different species. It's very difficult. But for plants, we can do this. We can. We have a hybrid between the potato and tomatoes. Yeah, we have some potato tomatoes plant, and we have the tomatoes on the upside of this plant, and near the roots we have the potato. Yeah, for plants it works, but for animals it doesn't work. But by using genetic editing technology, we can try to do it. Yeah. About is DNA restructuring after spider bite in Spider-Man? There are viruses that can insert regions of DNA into human cells. But what's the problem? Because, for example, we have a spider that bites us in the hand, and in the place when spider bite, we can change the DNA. May be changed, but DNA, we have the same DNA in all of our cells, and we need a special virus that can affect all of our cells, and our immune system must be stopped. And it's very difficult to me. It's a very complex thing, but it may happen. Yeah, viruses evolve too, and in distant future, maybe we have such virus that can go that can hide from our immune system, can go to our cells, change something in our cells, and not destroy our cells. Because what's the problem with viruses? Because when they change something, they can change some genes. Maybe we can change gene that creates ribosomes, for example, and we can create ribosome. The cell can create the proteins and died, and in the result, virus died too. It's very difficult. Virus need a very precision technology to add something to our DNA to do it. To use alternative AI algorithms. I think genetic algorithms. Yeah, it's more popular here, and classical learning. Deep learning, in some cases, is used, but we need more data. In biology, the main problem with biology, we don't have a lot of data because it's very difficult to extract from people additional samples. It's not so ethically, it's not so ethical to have a diseased people. We need to go to the tissues, remove something from it, and do it several times. I think it's cruel. Yeah, and we can do it. That's all. Yeah, if somebody have questions.
If nobody has questions, we can conclude our live stream. Thank you, Artem, it was a very extensive presentation. I did not expect there to be so much biology. I thought you're a fellow machine learning engineer, not just
I'm not machine learning, but I think it's interesting to talk about the biology, because machine learning, I think the most of people know about the machine learning. Yeah, that's why it's more interesting to talk about the biology.
Yeah, I'm personally in the medical machine learning field too. I'm doing computer vision for medical imaging. So for me, it's also really fascinating to talk about domain knowledge, about how the images are taken, how the data is annotated. But some engineers don't agree with me. They only care about models, and they don't care how the data comes to them. Anyways, it was really, really interesting from you. I think we can conclude here. So goodbye, Artem.
Yes, thank you.
There was another question. Could you elaborate on internship opportunity? How does it happen in Contori? But I think I can add to live stream.
Yeah, I can answer this question. This is connected with Contour Academy. As I said before, we have some external courses for students, for all interested people in acquiring new knowledges. So you can follow us on social media, because now I can't say you the exact dates of starting new schools, as we just closed the application form for Python and Query School, and these courses will be start soon. So we are going to start DevOps school, but I believe this will happen maybe in several months. This is not an internship, but this is a better way to gain new knowledges, practical knowledge, and to maybe get an opportunity to receive a job offer and continue working with Contori. This course is instead of internship. And also, for example, if you This is free of charge, and if you even don't get a job offer, you have a lot of knowledge, a lot of expertise, and you will mention in your CV that now you have practical cases, you know how it works in real life, not just by reading theoretical material. So we will speak more about what you should mention in your CV on Monday, because we invited our colleague who is HR manager to make for you HR workshop and tell you more about how to write your CV the most effective way and how to make it catchy, because we understand and we realize that you're students and you don't have any experience. Maybe you have, but not so much as others experienced developers. So we will talk about what to write, what to mention in your CV to draw recruiters' attention and to get a job offer. And also on Monday, we will discuss your projects after hackathon. Maxim, will you announce it later, or maybe in Telegram chat or by email?
Can you repeat, please? What should I announce?
I mean our Monday schedule.
Yeah, it was in the original schedule, and we'll do an extra announcement in our
Okay.
Telegram group, so it's okay.
Okay. So I believe if we don't have any questions, we can.
It's time to wrap up. Thank you, Anna. Thank you, Artem.
Thank you guys for joining us today.
Thank you.
Maybe don't you have questions about hackathon tasks?
I guess the questions will be tomorrow at our Telegram chat.
Okay. Please add us, Artem and me, to Telegram chat to answer the questions.
Yeah, I'll add you. So I think it's time to wrap up. So
Okay, so we wish good luck all participants of our hackathon, and I would remind that we have some prizes for our winners for the first, second, and third places. This is corporate branded, very high qualified merch of our company. This is bags for laptops, hoodies, and mockups. And we will be excited to present this for our winners. We wish you good luck, and if you have any questions, please type to me or Artem or send message to Telegram chat. And of course, follow us to stay tuned.
goodbye.
Okay, goodbye. Thank you
Let's say
guys.
bye-bye to our chat.
Have a good day. Have a good weekend. Goodbye.